Thank you very much for accepting my invitation.
Yeah, thank you. Thank you so much. Thank you. I've actually really enjoyed the day. I realize I'm normally really tired by this point in a visiting lecture. I'm not tired at all, so I think it's been a really, really interesting day. Yeah. So today I'll be talking about focus of my research program, which is really trying to answer this question: do we need biodiversity for ecosystem services? So certainly biodiversity loss and its impact on humanity is one of the great challenges of our time. And ecology as a field has taken a rigorous experimental approach to answering this question, setting up large manipulative field experiments like Cedar Creek or Jena, shown here, in which they experimentally assemble communities at different levels of species richness, in other words, with different numbers of species, and measure how the amount of ecosystem function, whether that's carbon storage or nutrient cycling or biomass production, how that changes with the number of species present, in other words, with biodiversity. And in setting up these experiments, they really focus on species richness, and then they control, in other words, to isolate richness, they control other aspects of ecological communities, such as the total abundance of individuals that you might have or the identity of the species that you have present. So Brad Carnelli of Michigan reviewed about 600 biodiversity ecosystem function experiments in Nature a couple years ago and came to the clear conclusion that in general, across all these experiments, they show that as you increase species richness, ecosystem function increases and then kind of asymptotes. But in general, this pattern is quite robust. In other words, biodiversity does increase ecosystem function for many different kinds of functions. So the question I'm interested in is, how do we scale up from these experiments to real-world ecosystem services? And I'll just make a definitional note here. By real world, I just mean large scale, and the scale difference here is pretty substantial. So biodiversity function experiments are typically measured in gallons or in square meters. My study systems are thousands of square kilometers. And I also mean unmanipulated, so natural, free-living communities. And then another definitional note, ecosystem services are technically the subset of all ecosystem functions that are important to human well-being. I tend to use the term function and service interchangeably because the service I work on is pollination, which is both a function and a service. It also just phenomenologically, people who are experimentalists tend to talk about functions, and people who work in real-world landscapes like me tend to talk about services. All right, so in this same Nature paper, Brad also reviewed the existing literature on biodiversity and ecosystem services in real-world landscape. And even though there were a fair number of papers, the conclusion was very different. The conclusion was basically, we don't know. So here's how Brad very tactfully put it: the evidence for the effects of biodiversity is mixed, and the contribution of biodiversity per se to the service is less well defined. All right, so why is it important to know how important biodiversity is to ecosystem services at these large scales? I think it's pretty important, largely because the conservation and policy worlds have pretty much moved ahead with the belief or assumption that biodiversity is very important. For example, the United Nations, the IPBES panels, which are sort of modeled after the IPCC panels, but on biodiversity, they are the panels on biodiversity and ecosystem services, and they basically treat these two things as, if not synonymous, very broadly overlapping. The first panel met in 2017 and actually used pollination as the ecosystem service that it reported on. And secondly, in the nonprofit world, this is Mark Tursek, who was until recently the CEO of the Nature Conservancy, and his book Nature's Fortune, which very strongly takes this perspective and makes this argument that, in fact, the reason to conserve biodiversity globally is because we need to have ecosystem services and we need biodiversity for ecosystem services. So this is sort of put into practice, even though the scientific evidence is not really there. All right, so the rest of this talk will be about how, in my research program, I'm trying to answer this question: do we need biodiversity for ecosystem services at large scales, and to explicitly scale up not just the data, but how we think about the question, because we sort of have to think about the question a different way in order to even address it at these larger spatial scales. So how I'm trying to do that between the biodiversity function experiments like Cedar Creek here, and then this image on the right, which is actually a Google Earth shot of my study system in New Jersey. All right, so the ecosystem service that I will be talking about today is crop pollination. Pollinators, animal pollinators, are required for about 75% of global food crops. So they're very important. Of course, the honeybee is a managed pollinator that's widely used, but substantial amount of crop pollination is also done by native wild bee species, and that's what I study as an ecosystem service. So in my study system, there may be 400 or so wild native bee species, and they are, depending on the crop, doing between sort of 10% and 70% of the crop pollination. So this is my method section. All the data I'll be showing you are collected in fundamentally the same way. So to measure biodiversity and pollination, we go out in the field and net bees that are actually visiting crop flowers. This is a commercial cranberry farm. And that is our measure of biodiversity. We bring these bees back to the lab and identify them to the species level. And then in a separate set of experiments, we take unpollinated flowers and present them to bees that are foraging on the crop and let the bee visit the flower just one time and deposit pollen like it normally would. And then we take that flower back to the lab and make a slide out of the stigma and count how many pollen grains were deposited. So that's the sort of function per flower visit. And then to estimate the total pollination provided by each bee species is basically just the product of the number of individual bees we found foraging on flowers. So that's like a count of flower visits from that bee species times the amount of pollen deposited per flower visit. And that's the ecosystem service from that species. So these methods allow us to actually separate the ecosystem service into the components that are done by each bee species in the system. All right, so the studies that I'm going to talk to you about today are getting at this question of how the biodiversity ecosystem service relationship is different in the real world because the real world is different from experiments in several ways. And these are sort of the big 4 ways that I think are probably most fundamental and that we try to study. So one is that in the real world, abundance varies dramatically across communities. Another is that species loss is not random. Experiments assemble communities randomly, but species loss in the real world is not random. Another is that real-world communities have very strong dominance in the species abundance distribution. In other words, there'll be a few common species that are really common, and then there's this long tail in the distribution of really rare species. And basically all ecological communities are like that. And then lastly, there's species turnover across space in the real world. So I'll just show a little bit more what each of these is like. So in the real world, you could have this diverse community on the left that's storing carbon and this not diverse community on the right that's storing carbon. And it's quite possible that the one on the right is actually storing a lot more carbon just because it has more individuals. This is a diagram that shows the point about non-random species loss. So this is showing the sort of traditional biodiversity ecosystem function experiment graph with species richness on the x and ecosystem function on the y. And experiments, because they randomly choose species for each level of richness, the relationship between species richness and ecosystem function would be expected to be linear, with the slope is just the mean contribution to function per species. That's sort of the expectation for random species gain or random species loss. In the real world, we're doing a really different thing. We're not randomly assembling communities, but we're non-randomly disassembling them. So we're starting in the upper right. And you could imagine 2 extreme scenarios. In one, if we lose all the most important species first, we'll be on curve C, and we'll lose function really quickly. Alternatively, if we lose the functionally unimportant species first, we could be on curve B and maintain most of the function until we get down to where we really have very low species richness. So that's sort of the possible space, I guess. And what actually happens in the real world is not well known at all. All right, third thing was dominance. So this picture just shows natural communities on the left, where you typically have one species that's, there are lots of individuals and then a few rare species. Experiments have mostly been set up like the picture on the right, where they establish communities that are more even. For example, if they seed in 5 plant species, they put an equal number of seeds per plant species at the start. So that's very different from nature. And then lastly, this issue of species turnover. So this is the only one of these ideas that is very explicitly spatial. It's about when you try to scale up questions about biodiversity and ecosystem services, how is just the fact that you're expanding over space fundamentally different? So in real-world landscapes like on the left, if these were 3 different tree communities and they were each storing carbon, in the first community, you need those 2 species to store carbon. Then in the second one, you have one of the same species, but now you need these 2 additional ones if you want to store carbon in both those communities. And in the bottom one, you're adding another species if you need to store carbon in all 3. So you get this sort of species accumulation curve just due to turnover of beta diversity across space. Now the experiments part, I left empty on that because experiments really can't investigate this issue of species turnover of beta diversity because the experimental communities are assembled by the researcher. So any turnover in species was just part of the experimental design. All right, so turnover should mean you need more species to meet your service when you consider larger scales. All right, so to sum up then our predictions from these sort of 4 basic principles, abundance variation in the real world might mean that species per se don't matter that much in the real world for ecosystem services. In other words, the importance of species might be overwhelmed by just the importance of abundance variation. Non-random species loss order could either increase or decrease the importance of biodiversity compared to in experiments. Dominance, I would expect to mean fewer species are needed in the real world because you might just need like the top few common species and the long tail of rare species don't do a lot. And lastly, species turnover should mean that you would need more species in the real world when you are looking at larger scales. So I'm sure you've noticed that these predictions are not all in the same direction. So there isn't really a clear overall expectation for what might happen in the real world. All right, so the first study that I want to tell you about was this paper here that we published in 2015. And we used the data on crop pollination that I was just describing to you for 3 crop systems. One is watermelon, that's the yellow flower, and then cranberry is that pinkish flower, and blueberry is the white flower. Their fruits are probably more familiar, but to pollination ecologists, the flowers are much more familiar. So to me, the most exciting thing about this paper was actually the analytical approach we used, which hadn't been used a whole lot in ecology at that time, and it made it possible to separate the relative importance to ecosystem services of species richness, just the number of species you have, species composition, the identity of which species you have, and very importantly, just changes in abundance, regardless of species identity. So that analytical approach is the price equation, which will be familiar to any evolutionary biologists, of course. So the price equation was developed about 50 years ago by the evolutionary theorist George Price. And he used it to partition evolutionary change between parents and offspring into additive components that were attributable to natural selection and various other things. So my collaborator, Jeremy Fox, created a form of the price equation that can be used for biodiversity ecosystem function analysis that partitions the change, in my case, the change in pollination between two sites, so it's fundamentally a pairwise thing, looks at, say, 2 farms in my data, the difference in pollination received at those 2 farms, and partitions that into additive components that are attributable to changes in richness and the number of species you have, changes in the identity of the species you have, and for the purposes of this paper, I'm sort of considering those jointly. They're additive, so I can add them. It's like all changes that have to do with species. And then the third one here at the bottom is just changes in abundance. So that means there's a species that was present at both sites, so it doesn't contribute anything to changes in richness or composition between sites because it was at both sites, but its abundance could vary a lot, and that does affect the amount of pollination, or could. All right, so this is not a statistical model. It's really just a descriptive tool, but it's super, super useful for what we're trying to do. So I'm just going to cut to the chase here with what we found here. So the title kind of says it. Abundance of common species, not species richness, drives the delivery of a real-world ecosystem service. So what we found is the effect size of all the species terms put together was only 15% as big as the effect size of abundance changes. In other words, it was these really abundance bees that they might vary 100-fold in abundance between sites, and it was that variation of these dominant species that really drove pollination, nothing to do with what species were present particularly. So what I want to spend some time on here is why we found that. Like, what is the mechanism underlying that result? And I feel like I can explain it pretty well just by showing you the data. So first, just a quick reminder of what we're actually measuring. It's pollination services per bee species. So like if these bumblebees are all the same species, we take the number of bumblebees, whatever that is, 8 individuals, times the number of pollen grains per individual. Say it was 4, we'd have 30 true pollen grains is the ecosystem service delivered by that species of bumblebee. So then what I do with that is plot what I call species ecosystem function distributions. So they're very analogous to species abundance distributions for ecologists who are familiar with that. And in fact, for my data sets, the species function distributions match onto the species abundance distributions extremely closely, like R of 0.9. But what I'm actually showing you here for each of these 3 crops, on the X is the total number of pollen grains deposited per species. These are just histograms. And on the Y is the number of species that deposited that number of pollen grains. So what you can see is that most species are not depositing very many pollen grains. There's a really tall bar at the left. And then a few species are depositing a lot of pollen grains. So these few species that are doing a lot of the pollination, these are also the highly abundant species. And conversely, these species, the majority of species that are not doing much pollination are the rare species. Often they're just represented by 1 or 2 individuals. Okay, so sort of what this all means for the importance of biodiversity to ecosystem services, the interpretation is quite similar for these 2 systems, the watermelon and the cranberry. It's a little different for blueberry, so I just want to talk about these 2 together first. So these very common species, these functionally dominant species, are present at virtually all sites. So they don't contribute anything to changes in richness or composition, but their abundance varies so much between sites that they actually drive the pollination. They drive the differences in pollination between sites. And in contrast, these rare species contribute little to pollination, but because they're so rare, they're not present at most sites. They kind of swap in and out among sites. They might be present at 1 out of 20 farms or something. And so the changes in presence and absence of these rare species is what drives species richness changes. But they don't do much pollination. So I'm going to pause slightly on this because I think this is really profound, actually. So the dominant species account for the pollination, but the rare species account for the species richness. And so due to the shape of this species abundance distribution, ecosystem services are decoupled from species richness. So that could be very general because all ecological communities have these very skewed species abundance distributions, and it is very different from what we think we know about nature from experiments. All right, so I said the blueberry was a little bit different. So it was different. It still has a very skewed distribution, obviously. It's different in the sense that there were more of these common species, rather than just one. And in fact, there was a pretty strong relationship between species richness and pollination for the blueberry system. But the reason that that relationship existed had only to do with these 9 common species. So like if you had one of these 9 common species, you did not get much pollination. If you had all 9, you got a lot. So species richness was important to biodiversity, but it was sort of 9 common species, which from a conservation point of view is kind of limited. All right, so I now want to tell you about a second paper that my lab contributed to that was published in the same year. This was led by David Klein of Wageningen University. And I think this figure really tells the story of the paper, so I'm just going to walk you through this figure, and I think you'll see the point of the paper. So this was a global synthesis. It brought together all the labs that had data on crop pollination, bee biodiversity, and visitation to crop plants, of which my lab is one, but there are quite a few, so that here on the x-axis is a number of studies. So it went up to 90 studies. They're from all continents except Antarctica. And in this one, we're just using visitation as the metric of pollination. Not all of these studies had actually counted pollen grains. But again, in the data sets we have that measured both pollination and visitation, the two are super highly correlated, like 0.7 to 0.9. So the x-axis is studies. This is just adding them up in all possible orders, right? The accumulation curve. The first line, the dark black line, is the regional species pool. So that reads over to the right-hand side there. And that is the total number of bee species that are known from all the regions where these studies were done. So that's sort of like the total pool of bees that could have been found on these crops, like they were in that region, so they potentially could have been contributing to crop pollination. So you can see that goes up to about 6,000 bee species, which is maybe a quarter of the bee species in the world. Then the blue dashed line are all the crop visiting species. In other words, those are the bee species that researchers actually observed on crops. That's obviously many fewer. It reads over to the left-hand axis, so it's about 600. So a tenth as many. And the reason that a bee would be in the region and we didn't record it on crops could be a bunch of different things, right? It could be the bee doesn't persist in agricultural areas. It could be the bee doesn't like that crop plant. You're running a pretty strong filter on bees when you study crops because it's just a monoculture of one plant species. It could be we didn't sample enough to observe it. It was there, but we didn't see it, right? For whatever reason, it's only about a tenth of them that are found on crops. And then the line here that's really the most important one is this third one. These are what we call the dominant crop visiting species. So these are the minimum set of species that were necessary to account for 80% of the flower visits on that crop in that study system. So that's saying, if farmers wanted to have 80% of their pollination and not have to conserve, all these thousands of bees, like which bees would they really need to conserve? And that is a really small number. You can see that thick blue line doesn't have much of a slope and doesn't even change that much as you add up all these different continents and crops and everything. It ended up being, I think, about 2% of all bee species actually made it into that dominant group. So the take-home that got a lot of media attention from this paper at the time was that 2% of bee species do 80% of the crop pollination. And hence the title, Delivery of Crop Pollination Services, is an insufficient argument for wild pollinator conservation. So that is definitely flying in the face of the idea that we should all be very motivated to conserve global biodiversity, because if we don't, we're going to lose all our ecosystem services, right? Okay, so the third paper that I would like to tell you about is one that we published last year, and in which I explicitly tried to do a couple things. So I tried to think about a way to scale up this question about biodiversity and ecosystem services in such a way that the analysis actually matched what I think people mean when they talk about scaling up biodiversity ecosystem function, which is sort of a jargony thing that, it isn't always clear what exactly is meant by that. The second thing that I tried to do is to explicitly separate the relative importance of dominance, which I would expect to depress the amount of biodiversity we need, and species turnover, which I would expect to increase the amount of biodiversity we need. Since these have contrasting predictions, I was curious about sort of the strength of those effects, in opposite directions. Okay, so in order to do this paper, I had to sort of transform what the question is when we talk about biodiversity and ecosystem services. When we're talking about now expanding over space, we're adding an additional axis. So we can't really just use the figure that everyone uses in the biodiversity ecosystem function experiment world, which is, they use this figure on the left here. So this is explicitly within one community, one set of plots. You have, sorry, this is within one plot. Think about it that way. So you have species richness, you have some number of species that you have, and then you have function that increases with species richness, and then you choose some level that is considered adequate function. Often people use something near the asymptote, and that level of function W, basically you read from W. In order to get that level of function, you go down and you say we need X species. So that's the way to do it when you're doing it at a smaller scale. When you're now asking this question across many communities, and this is what I think people really mean by scaling up, we want to know how many species do we need, not just to pollinate one farm, but 2 farms or 3 farms or 4 farms, each of them getting pollinated. So now this threshold level of pollination that's required is implicit in the graph. We have to just define that a priori. And then what we can plot is the number of communities that we're looking at on the X and the number of species that are required for pollination to be delivered to all these communities on the Y. So for one community, we would need X species, but we're now looking across all these different communities and doing an accumulation curve of how the number of species we need increases with increasing spatial scale. All right. So the study system that I used to answer this question are, again, these 3 same crop plants. I'm showing you here just the geographical version so that you can get a sense of the spatial scale. So that's New York up there and Philadelphia. My study sites are sort of arrayed in between there. The red are cranberry farms, the blue are blueberry farms, and the green are watermelon farms. And these are all commercial farms. So to sort of apply what I was describing there in terms of what the conceptual approach is, we're asking how many bee species do you need to pollinate one of these sites, and then to pollinate each of 2 of these sites and each of 3 of these sites, et cetera, just added up in all possible random orders. So I'm now showing you results, and these are the exact same axes as a conceptual diagram. These are just now showing you data. So there's watermelon, cranberry, blueberry, and then all three crops together. The thresholds, I did something that's often done in biodiversity ecosystem function work, which is to use a variety of thresholds because we don't really have a great answer for what the one best threshold is. So here I'm looking at 25%, 50%, and 75% of the mean level of pollination that was actually observed across all farms of a given crop. So it's scaled to what was actually observed. But you can see that all of these lines increase. They mostly seem to not asymptote with the possible exception of blueberry. In other words, if I'd kept sampling more farms, we probably would have needed more bee species. And also with this methodology, we're getting a really different answer from what we got in the two previous studies, where we had seen before only the abundance of the most common species is what matters for driving pollination. When you just ask what drives the difference in pollination between farms, that's the answer you get. With David Klein's study, we got the answer that you only need 2% of all bee species to do 80% of the pollination. Here, we get the answer that if you consider all three crops, so this one in the lower right, we're adding up to all 48 farms together, we actually need about 80 bee species, and the total number of species observed in the system is a dotted line at the top. So we had, I think, 110 bee species we observed, and if we want 75% of the mean observed pollination, if we want to keep that, we need to keep about 80 of our bee species, which is a really different answer. It's like 75, 80%. Okay. So other than that, I guess the sort of obvious point here is that if you want to keep a higher percentage of your pollination, you also need a lot more species. The curve on the bottom, that only maintaining 25% of the pollination, the reason that can be so much lower is, again, we have this very skewed abundance distribution where there are few dominant species, and just keeping a few dominant species can provide that lower level. The curves get a lot steeper when you increase the threshold. All right, so why does this particular analytical approach find that so many more species are needed compared to the other two? It's the same data. So there's obviously something different about the way that we're setting up the question. So I think the reason there are really two mechanisms here, and I can show them both to you by looking at the data in this figure, which I think is the most important figure for the paper, but this being science ended up in the supplementary material and probably no one's seen it. So this is what's really going on. So these are the three crops. The black line was the threshold. I think I'm using the 50% threshold here. And they're sort of qualitatively two different ways that species turnover could cause the number of species we need to have that steep slope and accumulate really rapidly as we add up across farms. And these are actually the bars here. Each bar is a site year. We studied these bars in multiple years, so we're sort of assuming the farmer needs pollination at each farm in each year as our unit. So the bee species here are colors, and they're stacked up in terms of the number of pollen grains that they deposit. And they're also stacked in order, so the dominant species are the first bar, which you can see are really big in a lot of cases. And then the rare species, the species that aren't contributing much, are up at the tips, all those short little stacks. And you can see we have a ton of bee species. I mean, those are our species list. So I said there were sort of qualitatively two ways that species turnover can matter here. One is that the identity of the dominant bee species actually turns over across farms. So like all these farms have strongly dominant species. No one farm may need a lot of species, but a different dominant species is needed at each farm. So when you add them up, you end up needing a whole lot of species. So that here would show up in these pictures as the bottom bar. The biggest bottom bar would be different colors in each of these columns, right? Like you'd see a lot of change in color. So you definitely see some change in color. But for some crops, it's often the same dominant, this sort of bluish, greenish, gray one, and watermelon and cranberry that's the dominant in both of those is Bombus impatiens, which is like the super common weedy trash bee in the east. So that's happening some. The thing that's happening more that is causing species turnover to really steepen those curves is that many farms are kind of close to the threshold, like they're barely making it, or they're even below it. And in that case, when you use a threshold system, you will, by definition, need all the species for that farm. Like these farms over here that are below the threshold, we need all those species, just by definition, even if they're very rare and they're not actually contributing a lot of pollination. So this is a very conceptually different approach to what sort of biodiversity and importance to ecosystem services means compared to the two approaches I was talking about earlier, which were based on proportions. If you're going to think about this question in terms of proportions, you will likely get the answer that the dominant species are most important, because by definition, dominant species are those that are the largest proportion of a community. If you use a threshold, you're in a really different realm. And I'm honestly not sure which way really makes the most sense for what people are trying to ask. Okay. So I want to point out a few more things about these curves. One is that the positive slope here, any positive slope we see, is due to species turnover. It basically can't be due to anything else. If they were exactly the same species every place, we would not get a positive slope. The other thing is that these are the slopes we observed. And these slopes are presumably flattened some by dominance, but we don't really know how much they're flattened by dominance. Like that is, they probably should be higher without dominance, but we don't really know how much. So to ask that question, we had to use a null model. So what we did is we can't change the natural communities that we have. They just have high dominance. These are free-living wild bee communities. They can't really manipulate them. But we can use the data that we collected, that's like literally our observed data, and just readjust it to make our natural communities more like experiments that have even species abundance distributions. So that's what we did. So what we actually did for each farm in each year, we had a list of species that we found there and the abundance of each. So we had an aggregate abundance. And in the null model, we used that same species list and the same aggregate abundance, but we just reallocated how many individuals went to each species to make the community perfectly even. So all we changed for the null model was making every site year a perfectly even community. So that was simulating the case in which we had natural communities the way that they actually are, with the exception that they did not have dominance. So they had instead these even communities to see how much that affected these accumulation curves. So the cartoon version of that looks like this. So here we have our same graph with the number of communities on the x and the number of species required to meet the threshold at all these communities on the y. And the observed would be black. That's what I've shown you so far that we plotted. And the blue arrows that go up from the distance between how many species you need at one site and how many species you need at n sites, that blue distance is due to species turnover. So that's like the effect size of species turnover in this graphical model. And then the red line is the null that we would run with these even versions of the communities. And the null we predict will be above the observed. We don't know how much above the observed it's going to be. But in terms of interpreting the null, that green arrow going down is the effect size of dominance, because we would have needed more species with the red null if we hadn't had dominance. We did have dominance, so it pushed things down by the height of the green arrow. So that's like my conceptual, graphical way of thinking about what we're trying to do. All right, so here are these exact same axes, and the red null and the black observed, and the blue and the green lines, and these are run with our actual data. And you can see that the effect of species turnover was much, much bigger than the effect of dominance. So it's really the fact that we have different species at different sites and in different years that is driving these curves. And if we hadn't had dominance, it wouldn't have made a really big difference, which sort of surprised me, actually. All right, so that's the bottom line. This is just the one run with all 3 crops, so you can see it's really a pretty dramatic difference. Okay, so what I've shown you so far to sort of summarize these 3 papers, the first study found that abundance fluctuations of the dominant species drive ecosystem services. The second found that 2% of bee species provide 80% of the crop pollination. And this was a global study. And the third found that 75% of the bee species are needed to provide a threshold level of pollination to 48 farms across about a 15,000 square kilometer area. So these are very different results. And in fact, these are different results that we used for the 2 studies that were only from my lab, like exactly the same data. The global one included our data. So at first, I thought that was a big problem. I actually sort of felt like it's part of my job as a PI of my lab to not let things like this happen, where you do analyses on the exact same data and get really different answers. And then I kind of made my peace with it, because I realized that really what my research program is about right now, like what we're really trying to do, is figure out how to think about this question. And in fact, running different ideas and different assumptions, basically, different ways of defining the problem on the same data is actually super useful. It sort of helped me understand why these different approaches are giving us different answers. And each of these approaches is valid. I'm very solid on that. They simply give very different answers. So that is sort of where I'm at with this right now. And I actually was just invited to speak at the World Biodiversity Forum in Switzerland in a couple months in a session at which, I will read this, the speakers provide bounded quantitative estimates for global consequences of biodiversity loss and other ecosystem service. So what we're all being explicitly asked to do is basically put error bars on our results, right? Like show biodiversity ecosystem services, analyses, projections, whatever it is we do, and put error bars on it. So I'm totally good with that. I'm a big believer in error bars. So I'm going to do that. But what I want to do, if I can actually get up the nerve to do this, is use the exact same data, all with error bars, and these different conceptual approaches, each of which I believe to be valid, and show that the difference in how the researcher defines the problem totally dwarfs the error bars. I mean, I'm pretty sure that the differences in how we define the question, the error bars are not even going to overlap, is my belief. So I'm hoping to do that. So that is actually everything I have. I want to thank funding, which mostly has been NSF and some USDA, 2 collaborators who've really played a very important intellectual role in a lot of the work I showed you today, Neil Williams and James Riley. And last but not at all least, 4 systematists, taxonomists, who've identified bee species, which I really believe that biodiversity research is only as good as its species IDs, and these guys are incredible. They've identified 120,000 specimens with us. So that's Sam Droege, Jason Gibbs, John Asher, and Joel Gardner. And of course, thank my really amazing lab group as it's changed over the years. So thank you very much. Yeah.
So first, I was trying to figure out the threshold you're applying.
Right.
Is it site-specific, or is it like, what are you saying?
No, it's not site-specific. And that's actually, we started out thinking it could be site-specific. And when we did that no model, we realized it gave some really weird answers and was like circular. I would love to talk to you about that at some point. But I mean, the threshold itself is based on the mean across sites. So the threshold is the same at each site. It is a number. It is not a different number at each site. So it's not like a proportion of what that site had. It is just based on the mean observed like number of pollen grains per site, basically. It's either 25%, 50%, or 75% of that mean. So that is unsatisfactory, to me anyway, in a whole bunch of ways. It's actually better than what a lot of biodiversity function studies do, because it's actually very tricky to find an appropriate threshold. The more common thing is to use 25, 50, and 75% of the maximum, which is totally sampling effort dependent, right? So I use the mean, which makes my stuff on the low side compared to a lot of other studies. It would be better to have a measure of, based on the biology of the plant, like what is the asymptotic level of pollination that this crop field needs. That's super, super hard to know for a whole bunch of complicated reasons, but it would be better.
And then my other question, I think you started to get into, I was wondering about, like a null model to get at the effect of this threshold
Right.
versus the turnover, and also about what part of the turnover is spatial versus temporal.
Yeah, exactly. So I originally wanted to look at 3 kinds of turnover: spatial, temporal, and among plant species, which is huge in the bee world because bees have their preferences, and plants bloom at different times a year. I had 3 plants in 2 years, so I had to give up my aspirations for looking at those 2 things, and I just focused on the spatial, for which I had 48 sites. So we did partition. I mean, the short answer is with 2 years of variation and 48 sites of variation across like tens of thousands of square kilometers, the space turnover was way bigger than the time turnover. And I honestly don't know what the appropriate unit is to compare a year to some distance of space. I'm sure you know some way to do that, but okay.
And what about the dominance? Can you get at that with a null? Like, I mean, the threshold. Sorry.
Yeah, so I don't know. The only, I got at the effect of the threshold through sensitivity analysis, basically, of which I showed you three levels, right? I mean, it gets trivial when you take it too high or too low, because it's a percentage of a mean, which is confined by what the data were. So I don't really know how to answer it other than that. I think people often they emphasize whether the curves asymptote more than like the number of species per se. Like the number of species is obviously sampling dependent anyway. But the curves were basically not asymptoting. Okay.
So I have always learned that the connection between biodiversity and species richness has a lot to do with like trophic redundancy, that like the decline of an abundant species can be mitigated. I'm wondering if you looked at that at all, if you looked at like how different species with a very similar ecological niche
Right.
change its effects.
Right, you mean like you lose one and then another one increases when the first one's lost?
It's like if a blueberry plant can be pollinated by like multiple possible species, but one happens to be overly abundant.
Right.
The less abundant ones change effects of decline.
Right. So I think you're getting at what people call the density compensation idea, which is usually formulated around competition, in the sense that the cause, like there's a competitive dominant, but then if it's taken away for some reason, like by the experimenter, these subdominants will then become dominant and fill that space and provide that function. So there is less evidence for that in nature than there is in experiments. Yeah. And I'm addressing that just out of my knowledge of the literature, because I didn't measure density compensation here. It's inherently a dynamic thing, or you need to do a manipulation. But particularly when you're talking about environmental gradients like human disturbance, the there tend to be sites that are really good for all species and sites that are really bad for all species. And there's some sites where there's just no bees because it's pavement in the extreme case. And there are other sites that are great for bees and there are lots of all species. And taking out one species has less of an effect, or observationally, the richness and abundance are very highly correlated often. So in experimental systems, a lot of the density compensation studies have been done in grassland plots, or particularly in aquatic microcosms, where they have a bunch of microbial things and they put in a food pellet every day. So there's a real resource cap. And in that situation, with constant environmental conditions and a fixed resource cap that's being drawn down every day, you get a zero-sum game, and those kind of competitive dynamics are super likely. In real systems, like the resource availability is in fact what's so different across sites. That's my belief about that, having not actually set up a study to study it. The other thing, and this concept gets sort of mixed with density compensation in the literature on ecosystem services a lot, but there's a different idea which I think operationally is very hard to separate, operationally meaning in observational data, which is called response diversity, which is a different species. It's not about competition, it's about niche, basically. The different species are differently adapted to different environmental conditions, so you need a lot of species so that one species is whatever, providing all the pollination in wet years, and then in a dry year, that species does terribly, and this other species that likes dry years is now your blueberry pollinator. And both, that goes on a lot in nature, I think. Yeah. Yes?
I was wondering if you know much about what kind of pollination benefits crop yields most. So, for example, you use like the number of pollen grains that come
Right.
from the flower, but I could imagine that the timing of pollination or like the identity of the plant the pollen came from could also matter.
Right. So actually, one of the reasons I like crop systems is they're super simplified, and some of those complications are, less salient, because you're absolutely right. In natural systems, actually, just whether you have conspecific or heterospecific pollen is like the 900-pound gorilla, right? I mean, there are all these plant species, and that's what really matters the most. In these monocultures, these are pretty, big monocultures of one plant, and we collect our data kind of out in the monocultures, so the heterospecific pollen thing is not really present that much. It's pretty much all the right pollen. And these are plants that are, bred to cross-pollinate, or in some cases even self-pollinate, but it still helps to have a pollinator. So I think they're about as well lined up with the assumption that it's just like accrual of pollen grains as possible. That still doesn't mean it's totally true, right? But like the distance to partners that are not closely related to you, like that goes away in crop monocultures too. Like all these real nature things kind of go away. But yeah, there could be a lot of this. I don't measure. And you go further downstream and measure like fruit weight would be the appropriate variable, since these are crops that are going to be marketed. It just introduces an incredible amount of noise, because like farmers don't water enough or they don't control pests enough, and it just becomes really hard to see a signal of pollination. Yes?
So you brought this up a moment ago, but during talk, I was wondering about what the role of biodiversity is outside of your system, where you're very focused on farms.
Yep.
But and I don't know very much about these guys, but in birds, people found that the birds are coming to plantations, stuff like that, often need nearby forests or
Absolutely.
habitat for breeding.
Yep.
And you mentioned that in some farms that are surrounded by concrete, what might be interpreted as better habitat to get
Right.
overall. So
Sure, definitely, yeah.
an argument for biodiversity in terms of sources of
Right. Right, and like land conservation to get just the pollinators you have now. Yeah, so bees are a little bit, there's a lot of evidence from systems other than mine that that natural habitat or semi-natural habitat, a lot of bees are kind of open habitat things anyway, so habitats like old fields are pretty good for them. There's a lot of evidence from other parts of the world that that is a big positive in terms of how much crop pollination you get. So the answer is definitely yes. In my actual system, it's kind of a flat line with respect to surrounding natural habitat cover, and that's kind of a different talk, actually. but yes, habitat, in agricultural landscapes, habitat availability can matter a lot, particularly when you're at the low end of having natural habitat. Yeah, definitely.
Okay, so I think that we're on time to finish. So
Okay.
thank you very much.
Thank you.